بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. First of all, I'd like to welcome all my dear students in semester 1 Mansoura Manchester program. I hope that all of you will have, inshallah, an amazing and happy semester. Here is Dr. Amani Abdel Fattah, lecturer of Histology and Cell Biology, Mansoura Faculty of Medicine. And today we will discuss together a simple and intelligent and interesting topic, which is the structure of the cell. It is the smallest unit in the living body. I think most of you hear about it before, and today we will summarize the structure of the cell and its components. Let's start first by the objectives of the lecture. So by the end of this lecture, all of you should know what is the definition of the cell and its main components, to identify the histological structure of different components of the cell, to recognize the functional significance of different components of the cell also, and lastly to correlate these organelles and the structure of these organelles to their functions. So here we have some objectives that we will cover in the lecture, inshallah. Let's start first by the question, what is the science that we will deal with? What is the histology? Simply, histology is the science that studies the structure of cells and tissues, and the study of these components or these small components of the living body need an instrument which magnify the picture of these components in order for our eyes to be simply seeing it. This instrument is called microscope. So simply, the science that we will deal with, which is the histology, it is the science that study the cells and tissues. Cells are the smallest units in the living body. When they aggregate together, they form tissues which is slightly larger in size. Here we need again the microscope for magnification, for enlargement of the photo to see the fine details of the cells and tissues. So again, our topic will be the structure of the cell. What is the cell? Let's start by its definition. It is the structural and functional unit of living tissues. Structure means that it is the unit from which the tissue is formed. Functional means that the function of tissue depends on the function of the cell. So group of cells of the same type do certain function of the tissue. So we consider it functional unit. So here the unit, the structural and functional unit of the living tissue, this is the definition of any cell in the body. Regarding the components, that is our topic today, any cell in the body have three main components. The first one, which is the cell membrane. This is the membrane. It is the membrane surround the whole cell and separate it from surrounding structures. Inside this membrane, we have cytoplasm. This is the cytoplasm, contain many organelles and inclusions, and the nucleus, which is considered the master of the cell or the big boss of the cell. So these three components together form the nucleus. In order to study the cell structures, so if we want to study the cell from inside, study the fine details and its components, we need a specialized type of microscope called electron microscope. Why this type specifically? Because this type have a high magnification power, so it can give us the smallest details of the cell. We can see it clearly by this type of microscopes. In this type, the components could be seen as pictures, printed pictures, or pictures on the computer. And these pictures are black and white like that. This is the picture for one of the organelles, which is mitochondria. So here, the picture that we get from electron microscope is a black and white one. In order to describe this photo or this picture, we have two terms that we will use frequently. The term electron dense means the black parts. So any black part like that, like this membrane, any black part of the photo, we describe it by the word electron dense. On the other hand, the white parts, the lucent areas, the faint parts, the pale parts, are described by the word electron-lucent. So again, in order to study the cell components, the very fine details of the cell, we use electron microscope that give us black and white pictures. In order to describe these pictures, we have two terms. Electron dense describe the dark parts or the black parts, and electron lucent to describe the white areas or the clear areas. This is a brief introduction about the study of the cell. Let's start by the components we mentioned now. The first component of the cell is cell membrane. Simply, cell membrane is the membrane surround the cell. So it's surrounded from outside and separate the cell from surrounding. In addition, it regulates the passage of materials into and out of the cell. So here, this is the electron micrograph of cell membrane. It is a black and white one, as we mentioned before. And here's cell membrane of one cell, this one. And here is the same cell membrane of another neighboring cell. So here we have two cell membranes, not one. This membrane surrounds this one, and this membrane surrounds this cell. This membrane is important for regulation, so it controls the passage of materials to enter the cell or exit the cell. When we have a picture electron micrograph by electron microscope, this abbreviation EM denotes electron microscope. So by electron microscope, cell membrane appear as formed of three layers. So we describe it by word tri-laminar. Tri means three, laminar means layers. So the word tri-laminar means that the cell membrane is formed of three layers. Here is one membrane again, and this is another one. So here are the three layers. We have two black lines, and in between we have white one. The two black lines, we will describe it by the word electron dense, yes. And the white one in the center or in the middle, we will describe it by electron lucent. So finally, the membrane is tri-laminar, three layers, outer and inner electron dense, black one, and the middle electron lucent, the light one. Here it's the same. This is another membrane of another cell, neighboring cell, and it is also formed of the three layers, the same layers, outer and inner electron dense layers with middle electron lucent one. Here is a higher magnification for the membrane, and here we can clearly see the three layers or the three lines. Here we have black one out on the outer surface, another black one on the inner surface, and in between we have white layer. Chemically, regarding the chemical composition of cell membrane, chemically the membrane is formed of the three components of any tissue or any living part. It's formed of lipids, proteins, and carbohydrates, but these components have a specific arrangement of them inside the membrane to allow it to do its all functions. We will not deal with the fine details. This will be discussed in the biochemistry or the physiology. But here we just mentioned that a cell membrane is formed of lipids, proteins, and carbohydrates have a specific arrangement. Let's check to the second component of the cell, which is the cytoplasm. So we mentioned the cell membrane. Second component will be the cytoplasm. Cytoplasm could be divided into three parts. So inside the cytoplasm, we have what we call cell organelles. And these organelles represent the living part of the cell. So they are living, they are functioning, they have a specific function that they perform. We have also what we call cell inclusions. And these inclusions represent the non-living part of the cell. So they represent the stored food inside the cell, for example. So one of the inclusions present inside the cell, the stored food. So this is non-living part. Both organelles and inclusions are suspended in a fluid. This fluid is called the matrix. So finally, cytoplasm could be divided into three parts. The organelles representing the living part, the functioning part of the cell. Inclusions represent the non-living or the stored components inside the cell, and both are present inside a fluid called the matrix. Let's start by the cell organelles, the living part as we mentioned now. Organelles will be subdivided into two categories or two groups depending on whether the organelle is surrounded or not surrounded by membrane. So if we have a separate membrane for the organelle, separating it from the other components of cytoplasm, we will call this organelle membranous organelle. So membranous means the organelle is surrounded by membrane. On the other hand, if the organelle has no membrane surrounding it, if the organelle is in direct contact with the matrix, we will call it non-membranous organelle. In each group, we have 5 organelles. Regarding the membranous one, it includes mitochondria, endoplasmic reticulum, Golgi Apparatus, Lysosomes, and finally peroxisome. All these organelles are considered membranous type. All these organelles are surrounded by membrane. In the other group, the non-membranous group, we have ribosomes, centrioles, cilia, microtubules, and microfilaments. So here you have 5 membranous versus 5 non-membranous organelles inside the cell. Here's a diagram for the whole cell. So here we have the cell membrane surrounding it from outside. Here we have different components of the cytoplasm, including the organelle, the matrix, the fluid in between, the inclusions, and here we have the nucleus, the master of the cell. Let's start by the membranous group, and let's have some little information about each organelle and its characters. The first membranous organelle is mitochondria. And in order to discuss any organelle in general, we have certain rules or certain points we need to cover. We will mention first the definition. What is the definition of this organelle? And simply we can write the definition without any study. It's simply the definition will include whether organelle is membranous or not, followed by a very brief hint about its function. So here in the mitochondria, for example, it is membranous or not, it's membranous organelle. What is the function of mitochondria? Is ATP production. So here is the definition. It is membranous organelle, provide the cell with ATP. Membranous organelle is the powerhouse of the cell. Membranous organelle give the cell its energy. All these definitions are correct. So in a very simple way, you can write the definition by writing the first part of the definition represent membranous or not. The second part of the definition will include the function of the organelle in brief, in a very short sentence. Regarding mitochondria, it's characterized by being numerous in active cells, and this is logic. Active cells need more energy, more ATP, more mitochondria. So we can find the numerous mitochondria in the liver cell, in the muscle cell. These are very active cells, and they include numerous mitochondria. By electron microscope, here we have a diagram. This is a not microscopic image, but just a diagram to illustrate the components of mitochondria. If we have electron micrograph for mitochondria, it reveals that mitochondria is formed of double-walled membranous vesicle. What does this mean? This means that the mitochondria is not surrounded by one membrane, but it's surrounded by 2. So here we have 2 membranes surrounding the mitochondria. We have this brown one and this yellow one. So we call it double-walled. Double-walled means it has 2 membranes surrounding it. Outer one, this one, as we see here, it is smooth. It has a smooth surface. This is the outer membrane of mitochondria. In comparison, the inner one is not smooth. The inner one shows projections, as you see here. These are projections. These are shelf-like projections getting out from this inner membrane into inside the mitochondria. And these projections, we call it cristae. So the inner membrane have projections called cristae. These cristae are very, very important inside mitochondria because it contains respiratory and electron transport enzymes. Enzymes very important for the ATP production, are present on this membrane. So again, we have two membranes, outer one which is smooth, inner one show projections called crusty. These projections have enzymes which are respiratory, electron transport enzymes. Inside these two membranes, we have a cavity, and this cavity is not empty. It contains a fluid similar to that of the cytoplasm. So we have, or we call it the same way. It's called the matrix, but here it is the matrix of mitochondria. It's not the matrix of the cell. So here, this cavity is filled with a fluid called the matrix. This matrix contains enzymes also, but these are Krebs cycle enzymes. In addition to, it contains DNA and RNA of mitochondria. So mitochondria have its own DNA and RNA, which is separate from that of the nucleus. So here are again the components of mitochondria. Outer membrane, which is smooth, inner membrane show crusty, and matrix inside contain enzymes of Krebs cycle, DNA and RNA of mitochondria. Here is the electron micrograph of mitochondria. So here we have the two membranes surrounding it, the outer one which is smooth, the inner show the crusty. These are the crusty projections from the inner membrane. And inside we have a fluid, which is the matrix. This fluid is granular. We can see it here. And here we have the electron dense granules inside the matrix. These are the three components of mitochondria by electron microscope. The second membranous organelle is endoplasmic reticulum. And as we mentioned before, what's the definition? Endoplasmic reticulum is membranous or not? Membranous, yes. So here it is membranous organelle. But here, because we have two different types of endoplasmic reticulum, we will not mention the function in the definition, because we have two different types with two different functions. But instead, we will mention that the shape is formed of branched tubules. Tubule is small, narrow tube. So the word tubule means small, narrow tube. So again, regarding the definition of endoplasmic reticulum, it is membranous organelle formed of branched tubules. As I mentioned now, we have two types of endoplasmic reticulum. We have one with rough surface, so we call it rough endoplasmic reticulum, abbreviated like that, RER. And another type which has smooth surface. Here we have the smooth type. We call it smooth endoplasmic reticulum, abbreviated like that, SER. So here we have two types of endoplasmic reticulum. The main difference up till now is the surface. One have rough surface called rough endoplasmic, and the other one have a smooth surface called smooth endoplasmic reticulum. In this table, we summarize the difference between the two types. So regarding the one rough endoplasmic reticulum, by electron microscope, it appears like that, parallel, flattened tubules. So the shape of rough endoplasmic reticulum by electron microscope appears like that, tubules which are arranged in a parallel form, parallel flattened tubules. Regarding the smooth type, the smooth is communicating narrow tubules. Communicating means they are branching and getting connected again, branching and connected again to form a network. So it appears like that, communicating narrow tubules. So here is the difference in the shape between the rough and smooth type. Regarding the surface, we mentioned that before. One have rough surface called rough endoplasmic, and the smooth one have a smooth surface. What is the cause of this roughness and this smoothness? Simply presence or absence of ribosomes. If the endoplasmic reticulum has ribosomes attached to its outer surface, so it is rough type. So the roughness here is due to the attachment of ribosomes to its surface. Regarding the smooth type, it is smooth due to absence of ribosome. No ribosomes are attached to its outer surface. So this is the second difference between rough and smooth type. Lastly, the function. Smooth rough endoplasmic, as I mentioned now, has ribosomes. So its function will be dealing with proteins. Ribosomes are the factory for synthesis of proteins, as you know. So here, the function of rough endoplasmic reticulum is to segregate. Segregate means separate. It separates the proteins synthesized by ribosomes attached to its outer surface inside its lumen, inside its cavity. Segregate the proteins synthesized by ribosomes inside its cavity. So it separates these proteins from the cytoplasm. In addition, it starts modification. Modification means addition or removal of certain parts of the proteins. So here, initial glycosylation. Initial means starting glycosylation, and glyco means carbohydrates. So glycosylation is addition of carbohydrates to proteins. This initial glycosylation is also achieved by the rough endoplasmic reticulum. So again, the function of rough endoplasmic reticulum is number one, segregate. And again, segregate means separate. Separate the proteins synthesized by ribosomes inside its lumen, so they are separate from the cytoplasm, the matrix. And in addition, it starts the modification of proteins by addition of carbohydrates, what we call glycosylation. This is regarding the function of rough endoplasmic reticulum. Regarding the smooth type, smooth will deal with the other components in the body. So here we have proteins. Smooth will have a function regarding the lipids and the carbohydrates more. So smooth endoplasmic reticulum is concerned with lipid synthesis, production of lipids. And in addition, it has a very important role in muscle contraction. And simply this role is that smooth endoplasmic reticulum in the muscle represents the storage site for calcium. So it stores the calcium inside, and the calcium has a very important role in muscle contraction. So regarding the function of the smooth type, number one, lipid production or lipid synthesis. Number two, have a role in muscle contraction, which is the storage site for calcium inside the muscle. This is regarding the two types of endoplasmic reticulum. Here we have a diagram for two types. Here we can see that rough endoplasmic is parallel tubules. Here we have the tubules, they are parallel to each other. And we have the ribosomes, these small dots represent the ribosomes attached to the surface of rough endoplasmic reticulum. On the other hand, we have the smooth type. It has a smooth surface, no ribosomes could be seen here. And the tubules are communicating, they are connected together, then become separate again in a random form, forming a network of tubules. This is the difference between the two types. Here we have two photos for the two types also by electron microscope. So who can tell me which of them is rough and which is the smooth and why? Number one is rough or smooth, and number two is rough or smooth and why? This one is parallel tubules, and here we have the dots attached to the surface, ribosomes. So this is the rough one, yes. In this one, we have cut sections, irregular, smooth surface, and smoothing, so they appear irregular like that. Here's the smooth type. So here we have a big difference between the shape of rough compared with the shape of a smooth endoplasmic reticulum. Let's check to the third membranous organelle, Golgi apparatus. Golgi apparatus is membranous or not? It is membranous type. What is the function of Golgi? Secretory function of the cell. So any cell have secretion, have secretory function, it will contain large Golgi apparatus because it plays an important role in the secretory function of the cells. By electron microscope, here we have a diagram first for the Golgi. Golgi apparatus is formed of three components. So here we have Golgi sac, microvesicles, and macrovesicles. These three components together form one Golgi apparatus. So here we have a diagram for one Golgi. Here we have the stack. These are the Golgi stack. These are the microvesicles. Micro means small. And here are the large vesicle, secretory vesicle. Here is the microvesicles also, or incoming transport vesicle. And this large one represents the macrovesicle. Regarding the Golgi stack is formed of 4 to 10 saccules. The word saccule means flat sphere. So if we have a bowl or a sphere and we compress it, this is the shape of saccule. Here we have 4 to 10 saccules arranged above each other like that. These saccules have two surfaces. One we call it mature phase, and the other one we call it immature phase. So here we have mature phase or transphase and immature or cis phase. Regarding the two types of vesicle we have a small one called microvesicle, and we have larger sized one called macrovesicle. The microvesicles, from its name, it's small. It will be present on or near to mature or immature phase. To be more suitable, it's mostly present near to the immature phase or the cis phase. And this microvesicle contain raw protein synthesized by the ribosomes attached to rough endoplasmic reticulum. So here in this type, microvesicles, they are small vesicles at the immature phase, and they contain proteins from rough endoplasmic reticulum. Regarding the other type, they are called macrovesicles. And these macrovesicles are present on the other surface, the mature phase. They are large and present at mature phase. And these vesicles contain proteins also, but here the proteins are modified. They are not raw. They are modified inside the Golgi stack, passing inside the Golgi stack to be modified by the enzymes present inside. And lastly, they get out from Golgi as modified proteins from as a mature phase, and they are called macrovesicles. These macrovesicles have two fates or two ways, whether to go to the cell membrane and get its product outside the cell as secretory product, like insulin, for example, like any hormone produced by the body or by any cell, secreted by cell, or remain inside the cytoplasm, and at that time, it will be converted to have the name of lysosome. If it remains inside the cytoplasm and contain hydrolytic enzyme, at that time, they will be called lysosome. So this is regarding the components of Golgi. We have Golgi stack arranged above each other. We have micro or small vesicles contain raw protein coming from rough endoplasmic reticulum. This protein will enter the Golgi, modified inside, and get out from the other surface as macrovesicle contain modified protein. This modified protein have two fates again, whether to go to the cell membrane and export its components outside the cell as secretory product, or remain inside the cytoplasm and have the name of lysosome. Here is the shape of Golgi by electron microscope. So here we have the Golgi stack, saccules arranged above each other. We have microvesicles, small vesicles, and macrovesicle, which are the larger vesicles. These are the three components of Golgi apparatus. Next organelle, which is in the membranous group, is the lysosomes. Lysosomes are membranous organelle, and these lysosomes are characterized by having a group of enzymes called acid hydrolases. These hydrolases are hydrolytic enzymes. They destroy components inside the cell into the simplest form. So acid hydrolases are present inside the lysosome for intracytoplasmic digestion. So it represents the stomach of the cell. So if we have a stomach to digest the food that we take, the small cell have a small stomach called lysosome. This lysosome contains enzymes for digestion inside the cell. These are called suicide bags, and here this term denotes that the lysosome can destroy the cell if it exports its enzymes outside. If the hydrolases get out from this membrane into the cell, it will destroy the cell itself. So they are called suicide bags. We have two types of lysosomes, what we call primary lysosome and secondary lysosome. These two types are two stages, we can consider that. So the primary in certain stage will be converted to secondary. But we need to know what is the difference between the two types. The primary one is small in size, compared with the larger secondary one. Both are spherical in shape, rounded in shape. So both are rounded vesicles, but the primary is mostly smaller in size. Regarding the content inside the core inside the lysosome, primary is characterized by having homogeneous core, and the word homogeneous means that all the components inside the primary lysosome having the same degree of coloration. We have no dark part and small and the paler part. All the color inside the primary one is homogeneous, the same degree of color. While in the other hand, the heterogeneous type, the secondary type, having heterogeneous core means that we have areas darker more than areas, denoting we have different components inside the secondary type. The last difference between these two types is the function. Primary lysosome is a fresh one, freshly formed lysosome getting out from the Golgi and not engaged yet in function, in digestion. So it's primary lysosome not yet started its function, not yet started the intracytoplasmic digestion. If primary lysosome ingests a molecule and starts the function of intracytoplasmic digestion, at that time we will call it secondary type. So this is the cause of that I mentioned that these are two stages. So primary will give secondary one if it starts or engaged in intracytoplasmic digestion. This is regarding the difference between the two types of lysosomes. Here we have electromicrograph for both types. So this one is primary one, and here we can see that it's surrounded by membrane, trilaminar membrane, and inside all the core, all the areas inside are having the same color. We have no areas darker than areas like that, but here the primary one is homogeneous. Compared with the secondary type, secondary lysosome have heterogeneous core, so we have darker areas more than paler areas. Regarding the secondary type, it is subdivided into two types. Secondary lysosome include heterolysosome or autophagosome. Both are secondary, both starting digestion. But what is the difference between? Hetero means that it is originating from primary lysosome, but this primary one fused with solid phagosome. Phagosome means foreign body. It is not a part of the cell. It is about material getting inside the cell from outside. So the word phagosome is a foreign body entering the cell. If this foreign body fused with primary lysosome, so the net result is the production of a secondary lysosome called heterolysosome. On the other hand, if the lysosome, the primary one, fused with part of the cell itself, like dead organelle is not functioning yet, the cell wants to discard it or to get rid of this organelle, this dead one and not functioning. At that time, primary lysosome will fuse with the dead organelle, and the net result is the production of secondary lysosome called autophagosome. So the main difference between hetero and auto is the content. If it contains foreign body, it is heterolysosome. If it contains part of the cell itself, which is not functioning, this is the autophagosome. So here, both originating from primary lysosome fused with foreign body with phagosome, it is heterolysosome. Fused with part of the cell, and this is the cause of calling it auto. Auto means self. So it ingests part of the cell itself. It ingests part of the cytoplasm, represented by dead organelle. Any part of the cytoplasm which is not yet functioning, the cell will discard it or remove it by being fused with primary lysosome to be digested. This is the fate of any dead organ. So here are the two types, the two differences of the secondary lysosome. Regarding the non-membranous group, we will start by the ribosome. Ribosome is a non-membranous organelle. It is non-membranous, means not surrounded by membrane, and as we all know, it is very important organelle in the production of protein synthesis, production of proteins. So It's non-membranous organelle, important for synthesis of protein. By electron microscope, if we have a photo for the ribosomes, these ribosomes are electron dense granule. Electron dense means black or white? Black. Here we have these black dots. Black dots represent the ribosomes. They are black or electron dense. We will not call it black, but we will use the term electron dense. Granules means dots. These dots are formed, each dot, by more higher magnification, it's formed of two subunits, large one and smaller one. These two subunits together forming one ribosome. And these two subunits are chemically formed of rRNA, ribosomal RNA, and protein. This is the electron micrograph for the ribosome. They are electron dense granules formed of two subunits, large and small one, and both are formed of rRNA conjugated with protein. Sometimes we see like that, we see a very thin ribbon or very thin filament of rRNA, messenger RNA, connecting different ribosomes or multiple ribosomes. In this situation, we call this whole structure polyribosome. Poly means multiple. Polyribosomes means multiple ribosomes attached to messenger RNA filaments. So these are connected together by a filament of messenger RNA that they are translating the code that it carries. Here, at that time, we call it multiple or polyribosomes because they are attached together to the same filament of messenger RNA trying to translate it. This is what we call polyribosomes. Regarding the types of ribosomes, we have two types of ribosomes. We have free ribosomes, and on the other hand, we have attached ribosome. This one, we mentioned that before. Who can remember where we mentioned the attached ribosome? Attached to what? Who can remember? Attached to the membrane of rough endoplasmic reticulum. Yes, these are the attached type. They are the ribosomes attached to the rough endoplasmic reticulum membrane. So here in the other type, free means it is not attached. They are free in the cytoplasm and not attached to any membrane. So here the free type, they are scattered in the cytoplasm. Regarding the attached type, it's attached to the outer surface of rough endoplasmic reticulum. Actually, both types are synthesizing proteins, but in the free type, the synthesized protein or produced protein used inside the cell, used by the cell itself. They are not producing protein to get out of the cell. But regarding the attached type, they produce protein for export, for secretion, or produce protein as lysosomal enzymes, which are present inside the lysosome. So both secretory proteins and lysosomal enzymes are produced by attached type, while the other proteins used by the cell itself for building its components, for building its membranes, these are synthesized by free ribosomes. Here is an electron micrograph for ribosomes. So here we have the rough endoplasmic reticulum with attached here. These are the attached ribosomes attached to the surface. And here we have free ribosomes in between, which are freely scattered in the cytoplasm. So here is the free type, and here is the bound or the attached type. Let's shift to the next non-membranous organelle, which is microtubules. Microtubules, micro means small, tubules is small tubes, so they are pipe-like structures. Pipe means tube, pipe-like structure of unfixed length, so its length is not fixed. It may be long, may be short, but its diameter is fixed. So all microtubules have the same diameter, but have different length. Here is one microtubule. This is one, it's a pipe tube with a lumen in the center. Here is the length. The length may be short, may be long, but the diameter, as you see here, it is fixed in all microtubules in the body. Regarding its structure, the Smallest unit in the microtubule is called tubulin. So here is tubulin molecule. It is molecule formed of two parts together are tubulin dimer. Di mean two. This tubulin dimer are arranged like that beside each other, forming a filament. This filament is called protofilament. So here tubulin protein. By polymerization, polymerization means they are arranged side to side, side by side like that to form one filament. This one is called protofilament. This protofilament, when arranged in a tube manner like that, 13 protofilament, 13 protofilament in a cylindrical form, they form one microtubule. So again, tubulin proteins or tubulin dimers arranged head to tail like that to form one protofilament, 13 protofilament arranged in cylindrical form, arranged in like a tube. This tube is called microtubule. This is regarding the structure of microtubules. By electron microscope, when we have a photo for microtubules, it appears like a tube, but it's very small, so we call it tubule. Hollow tubule. Hollow means it have a cavity in its center. It have a cavity like that, empty area in the center. It's not full. And its wall is formed of 13 protofilaments. So if you count these circles, each circle represents one filament, one protofilament. So here we have 13 protofilament in the wall of the microtubules. Here is another picture. Here we have protofilaments, 13 one, arranged beside each other in a cylinder form to form one microtubule. And here is the tubulin dimer, two molecules together connected to form one tubulin dimer. What is the function of these microtubules? Number one, it represents the cytoskeleton. As our bodies have a skeleton to keep our shape, also the cell have a skeleton. Cyto means cell, skeleton means the part of the cell that maintains its shape. So here in the microtubules, they represent part of cell cytoskeleton that keep the cell shape and protect it also. Number two, they represent the structural component of other organelles like centriole, cilia, and flagella. So the three organelles are formed of or part of their structure is microtubules. So it's an organelle sharing the formation of another organelle. Microtubules share in the formation of centriole, cilia, and flagella. Number three, its function also is important in cell division as a component which is very important that's produced during the cell division process is what we call mitotic spindle. This mitotic spindle is formed of microtubules. So the mitotic spindle is formed during cell division. Its component is microtubules. They are arranged in a specific manner to form the mitotic spindle. So here are the function of microtubules, represent cell cytoskeleton, structural component in building of centriole, cilia, and flagella. And number three, it's important for the production of mitotic spindle during the cell division. This clinical note means that the scientists get benefit from that the microtubules share in the formation of the spindle as they produce certain drugs, what we call cytotoxic drugs. These cytotoxic drugs are used for treatment of cancer. And the idea of these drugs is that they stop the formation of new microtubules. So indirectly, they stop the formation of mitotic spindle. So also as a result, at the end, they stop the cell division. So they stop the mitosis on the cancer cells, controlling the cancer cell development or the cancer cell division. So here, a clinical application for the microtubules is that there are a group of drugs, what we call cytotoxic drugs, used for treatment of cancer. Why they are used? Because they prevent the assembly or the formation of microtubules, so prevent the formation of mitotic spindle. So at the end, they stop the cell division, controlling the division of cancer cells. This is regarding the microtubules. Next organelle of non-membranous group is centriole. And centriole is non-membranous organelle which has a very important function during cell division. So this is one of the important functions of centriole. And in addition, it shares in the formation of cilia by formation of what we call the basal body of cilia. So it acts as a basal body of cilia, means that the basal body, which is part of the cilia, is formed of centriole. This is regarding the definition. By electron microscope, each cell contains two centrioles. Two. So it has a pair, one pair or two centrioles. They appear like two short cylinders, two tubes, perpendicular to each other. So you have an angle of 90 degrees in between. They are perpendicular to each other. Its wall, the wall of each one, the wall of each centriole, each one of those, is formed of 27 microtubules. So here, if we count the number of these tubules in its wall, we'll find them, they are 27. These 27 microtubules are divided into 9 groups: 1, 2, 3, 4, 5, 6, 7, 8, 9. And each group is formed of 3 microtubules. So we can call them 9 triplets. Try again 3. Triplet is a group formed of 3 microtubules. So here in the wall of centriole, we have 27 microtubules arranged in 9 peripheral triplets. Triplet means 3 microtubules together. The same here. Here is the one centriole, and here are the 9 triplets in its wall. Here is the same in another diagram. LS section, longitudinal section. So here are the 3 microtubules, triplets. We have 9 triplets in the wall of centriole. And this is a cross section or transverse section. Each circle from these 3 circles represent 1 microtubule. Here we have 3, 1 triplet, and here we have 9 triplets in the wall of centriole. Is by electron microscope. So here is the triplet. This is 1 triplet. 1, 2, 3. 3 microtubules together forming 1 triplet. And here we have 9 bundles or 9 triplets in the wall of centriole. If you have, or if you notice this circle, you will find that the wall is 13 protofilaments. This is a microtubule, and these small light dots represent the 13 protofilaments forming the wall of each microtubule. Next organelle is the cilia, and here is the shape of cilia. They are long processes. They are hair-like, long and projecting like hair from the border of the cell. So here is one cell. This is the free border of the cell, and these are the cilia projecting from its surface like our hair. As our hair projecting from our scalp, here we have cilia projecting from the cell. They are hair-like processes projecting from the free surface of certain cells. Where we can see cilia in the body, we can see it in the respiratory tract, in the female genital tract. These are examples of sites of the body having cilia. Regarding their number, they are hundreds per cell, several hundreds per cell. So each cell, each ciliated cell have hundreds cilia projecting from its free border. The function of cilia is that they beat in one direction. So all the cilia move together in one direction, and this helps the movement of a film of mucus or fluid on its surface. This occurs, for example, in the respiratory tract. The movement of cilia help getting out of any foreign body, getting out of mucus from the respiratory tract, and they beat in the direction of the outside the body, so it can, or it help getting rid of any mucus, extra mucus, or foreign bodies from the respiratory tract to get outside the body. This is the function of cilia. They beat in one direction, moving a film of fluid, and this film to which any foreign body can stick, so it help getting rid of any foreign material or mucus from the body. This is regarding the cilia. By electron microscope, cilia is very similar to the centriole. As we mentioned before, centriole form the basal body of cilia. So here, if we have one cilium like that, cilia is divided into shaft. Shaft represents the part projecting from the surface or projecting out of the surface of the cell. Here's the cell membrane. So here we have what we call basal body next to the cell membrane directly inside the cytoplasm. And here we have rootlets, which are like fibers projecting from the cilium into the cytoplasm of the cell. This shaft is the projecting part outside the cell. Basal body is the same as centriole as we mentioned. So here we have 27 microtubules arranged in 9 triplets in the basal body. But in the shaft of cilium, it is different. Shafts of cilium contain 20 only, 20 microtubules, not 27. And these 20 are arranged in 9 groups also. But here the number will be doublets, not triplets. Doublets means 2. So each bundle is 2 microtubules, and we have 9 bundles. 9 multiplied by 2, so we will have 18 microtubules. So the whole number we mentioned that it's 20. Where are the remaining 2? They are present in the center, and they are separate. So they are called 2 central singlets. 2 central separate microtubules. They are not attached like that. They are separate. So we call them singlets, not doublets. So again, the shaft of cilium contain 20 microtubules only, not 27. And these 20 are divided into 9 peripheral doublets, 9 doublets, and 2 central singlets. Why they are called singlets? Because they are 2 separate from each other, not attached like the doublets. This is the wall of shaft of cilium compared with the wall of centriole. In the basal body, it is typical centriole. It is 27 and 9 triplets. But in the shaft, it's different. They are 20 arranged in 9 doublets and 2 singlets. Here's the electron micrograph. And here we can see that we have doublets, 9 doublets peripheral, and 2 central singlets in the center of the shaft of cilium. Cytoplasmic filaments are the remaining non-membranous organelle, and from its name they are filament, and filament means thread. So they are thread-like structures. Thread-like structures mean that they are very thin and not having a cavity in the center like the tubule. They are just a filament. We have 3 types of microtubules according to its diameter. Sorry, 3 types of microfilaments. According to the thickness or the diameter, we have thin, intermediate, and thick. So here, from its name, we can detect its diameter. So we have thin type, and we have thick type, and in between we have intermediate type. Here we have the section of the diagram for the cell. These tubes are microtubule. It have a cavity. So this is a tube. It is microtubules. And here are the microfilaments. They are thread-like filament, not having a cavity in the center and forming a network inside the cell like that. These are the microfilaments. These are the intermediate type. They are a part of the filament or another type of filament. This table summarizes the difference between the 3 types. So here we have thin, we have intermediate, and thick filaments. First, we will mention an example of each type. In the thin filaments, we have actin, and the actin filaments are one example of thin type. On the other hand, in the thick type, we have myosin. So actin and myosin are 2 examples, one for thin filaments and the other for thick filaments. Regarding the intermediate filaments are different types or different groups. We have 5 types of intermediate. One of them, or just one example, is called the dysmin. So this is example of each category. Actin for thin, myosin for thick, and dysmin for intermediate. The second point is the contractility, whether the filament is contractile or not. Actually, thin and thick are contractile. So both could be found in the muscle, and they share in the capability of the muscle to contract. So thin filament and thick filament are contractile filaments and are seen in the muscle. While intermediate, they are non-contractile, so they are not sharing in muscle contraction. Regarding the site and function, we mentioned that thin and thick, the first site in the body is the muscle, because they are contractile and the enable the muscle to contract and relax. So the first site for thin filament is muscle for contraction, and also the first and the most important site for the thick filament is the muscle also, because together with actin, they are responsible for muscle contraction. In addition, we can find actin in other areas, like the microvilli. They are small projections projecting from the inner surface or the cells of the small intestine and large intestine. And actin present here, enabling microvilli for very fine movement. Also, actin could be found in the cleavage furrow, and this cleavage furrow is the area of separation of two cells after cell division, and the area of separation of the two daughter cells to give finally two daughters separate from each other. This is the cleavage furrow and it's formed of actin. When this actin contracts, it divides the cell into two daughter cells. These are the sites for the actin. Regarding myosin, it's mostly found in the muscle. Other than the muscle, it's rare to find myosin filaments in the form of formed myosin filaments. They are in the form of filament, mostly in the muscle, not more than that. Regarding intermediate type are diverse and could be found in different body tissues like the muscle, but in the muscle they are not sharing in muscle contraction. Remember that they are present as a structural component, not sharing in the contraction. Also, they are found in the nerve cells and in the epithelial cells, and in these sites, it represents part of the cytoskeleton again. So, intermediate filaments together with microtubules represent the cell skeleton, the cytoskeleton for the cell that keeps its shape. Here are the filaments and the differences between. Here is the diagram for the three types or the cytoskeleton. Here we have the intermediate filament that together with microtubules together represent the cytoskeleton, and here are the actin filaments, the thin type or an example of thin filaments present inside the cytoplasm. By electron microscope, here we have part of the muscle, and this is what we call the sarcomere. If you hear about it before, the thick lines, black lines represent the myosin. It is a thick filament. Very thin, faint lines here represent the actin filaments. Here and here also, they are thin and paler than the myosin, which is thick and appearing electron dots. Interdigitation or together form the port or the sarcomere, which is the structural and functional unit of the muscle. This is regarding actin and myosin filaments by electron microscope. Let's shift to the second component of the cytoplasm as a whole. So you mentioned all the previous data representing the membranous, the living component of cytoplasm, which is the organelles. Second, the component of the cytoplasm, which is cell inclusions. They are the non-living part, as we mentioned before. It represents the part which is not functioning, is not having an active process inside the cytoplasm, but it represents the stored materials inside the cytoplasm. These stored materials may be stored food. If the cell have extra food that is not in need to be used at that time, it's stored for the times of need. Like extra carbohydrates or lipids could be stored inside the cytoplasm for the times of need to be used by the cell later. Also, example for inclusions is the pigments. Some cells in the body contain pigments, and pigment mean colored substance. This pigment may be exogenous, so we take it from outside, like the dust that can enter the body with the air inspired, like the minerals, like the tattoo, which is the injection of dye under the skin. All of these are examples of exogenous pigments enter the cell from outside. Other types, here is the example for the tattoo, which is the injection of pigment under the skin. This is example of exogenous pigment. Inside the body, we have endogenous pigments that are synthesized inside the body, like the hemoglobin. This is a pigmented material present inside the RBCs, and it is synthesized by the body, so we consider it endogenous pigment. Endogenous means internal, not getting it from outside. It's synthesized inside the body. Or the melanin, which is the pigment representing or the cause for the coloration of our skin and hair. The presence of melanin is the cause for the coloration of our skin and our hair. It's black, or brown, depending on the pigment melanin present inside the cells. The last component of the cell after finishing the cell membrane, the cytoplasm, is the nucleus. And the nucleus represents the master of the cell, the most important component which control all the activities inside the cell is the nucleus. So simply, the nucleus represents the master of the cell, the big boss that control all the cellular metabolic activities and play a very important role in cell division and heredity because it contains a very important material which is DNA. DNA is important for cell division, helping the cell to divide and getting more number of cells, and in heredity as it carries the genetic informations of each body or each individual is written or is coded on the DNA. So again, the nucleus could be considered in some books considered an organelle, but for its important function, we separate it as a separate category or separate part of the cell, representing the master of the cell, not like just an organelle like the other organelles. It's present in all body cells except the mature RBCs and the platelets. These are the only two cells present in the body having no nucleus: mature red blood cell or red blood corpuscles and the blood platelets. These are only two cells having no nucleus. Other than these two cells, all the body cells having nuclei. By electron microscope, the nucleus is formed of nuclear membrane surround the nucleus, separating it from the cytoplasm, chromatin, which is representing the material from which the chromosomes are formed, nucleolus, which is a very small structure similar to the nucleus, so-called nucleolus, present inside the nucleus, and lastly, the fluid filling the spaces between the chromatin and nucleolus, and this fluid is called the nuclear sap, similar to the matrix of cytoplasm. These four components together represent the components of the nucleus. Here is the shape of the nucleus by electron microscope, and here we can see the membrane. This is the nuclear membrane. Its membrane surrounds the nucleus, separating its components from the cytoplasm outside. Inside the nucleus, we have nucleolus, electron dense part present inside the nucleus, chromatin, the gray areas. This chromatin represents the material from which the chromosomes are formed. And in between the electrolucent parts, the white parts, these are the parts of the nucleus filled with nuclear cell. These are the four components of the nucleus. Here is another photomicrograph for the nucleus. Here we can see also the membrane. You can see the nucleolus, the chromatin, which is the electron dense granules, and the sap, the fluid inside, which is representing the electrolucent areas or the white parts. These are filled with the nuclear cell. This is regarding the structure of the nucleus. So here I finish the lecture. I hope that it's clear for all of you. I hope that you understand it well, and waiting for any question of you, inshallah, maybe on the TA or in the group or in the channel, my YouTube channel on the YouTube. It's called Histology World, and I'm waiting for all of you to subscribe and enter to see more lectures. I will publish, inshallah. Happy to be with you today and hoping to see you later. Assalamu alaikum.